The overall goal of this procedure is to quantify the relative changes in the level of a protein at centrosomes under various conditions, for example, at a specific point in the cell cycle after inhibition of the proteasome. This is accomplished by first treating cells that are growing on cover slips with either the proteasome inhibitor Mg115 or with the control solvent DMSO, then immediately adding BRDU to both cells and incubating them for 4 hours in a cell culture incubator. The second step is to transfer the cover slips to a 24-well plate to fix the cells. Next, the cells are stained with antibodies against BRDU and the proteins of interest, and then with corresponding secondary antibodies. The final step is to acquire images of BRDU-positive cells using a fluorescence microscope and digital imaging software to apply identical exposure time and z-axis length to cells from each sample, then analyzing the images to quantify the relative level of centrosomal VDAC3. Ultimately, quantitative immunofluorescence microscopy is used to show that the centrosomal pool of VDAC3 is increased by 2-fold when the proteasome is inhibited during S phase.
The main advantage of this technique over existing methods like flow cytometry, immunoblotting, or quantitative microscopy is that this technique analyzes the relative centrosomal level of the protein in 2 differently treated samples that are processed on 2 different cover slips. This method uses normalization to an internal standard to control for the need to process the 2 experimental cell populations separately.
Though this method can provide insight into the regulation of specific proteins at centrosomes, it can also be applied to other situations, such as other subcellular